My name is Amir Karger, and I come from Harvard Medical School. So the basic question that I had, along with my colleague Victor Luria and a number of other collaborators, is pretty simple. How do novel protein structures and functions appear in genomes? So the early models suggested that it was really too improbable for intergenic DNA to mutate randomly and out would pop a protein. And so what they suggested instead is that new proteins would come from tinkering with other domains. And actually many mechanisms that we know of do this kind of thing. So they'll shuffle around exons that already exist, or they'll copy genes and generate new genes from them. But more recently, researchers have found, it looks like this is advancing without my asking it to, researchers have found many genes that actually did evolve from intergenic DNA. They've got homologs in the intergenic DNA of other species where they're not coding. And so we call these de novo genes. And we wanted to ask questions about de novo genes, namely, how many are there in a given genome? What types of proteins do they make? And then as they get older, how does that change? So we are looking at this in three complementary ways. First, mathematical modeling. So we built a statistical model that predicts the probability that these de novo genes will arise in the first place. Then we built a dynamic model that actually estimates how many you'll get in a genome. The second method is bioinformatics. So we're using Burkhard Rost and Chris Sanders' Predict Protein suite, as well as other tools, to look at many thousands of new and old genes. And we're also looking at translated intergenic DNA, since we believe the de novo genes recently were intergenic DNA, so they ought to be similar, but we can see how they're similar and different. I'll show you that on the next slide. And then also translating just random DNA and seeing what we get out of that. And finally, we're going to go into the lab. We're currently producing representative new and old proteins, and we're going to study some of their biophysical properties and ask, make sure that we can confirm that our bioinformatics predictions are correct. So our model said that many de novo genes could actually be created in relatively short evolutionary time, but that most of these de novo genes would quickly be lost to mutation. And actually our results show this. If you look at the human genome, for example, we have many hundreds of genes that are not found and homologs are not found in any other species. And then there's only a few that you find that are found in a few other species. And then if you imagine that you'll be saving a few genes every million years or so, then over time, once you go back a billion years, you'll find that there are many genes that have homologs in many different phyla. So now if we start asking what kinds of proteins we can make out of these, what these de novo genes will make, so if you translate intergenic DNA, and you ask how many predicted protein binding sites will there be, you find that there are actually a whole bunch of predicted binding sites, many more than you'll see with the older genes. And similarly, with the newest genes over here, you can see that, oh, you can't see this. Sorry about that. Do we have a pointer? We don't have a pointer. Sorry, very quickly. So with So over here we have the intergenic DNA. It's not working anymore. Thank you. Sorry about that. I was trying to use my mouse. It didn't show up. Bummer. So intergenic DNA, if you translate it, you see a lot of protein binding sites. The oldest genes, you'll see very few binding sites. And the newest genes over here, you see a lot of binding sites. So it's pretty similar to the translated intergenic DNA. And it's actually useful for new genes to be promiscuous, as we say, because that way they can try and integrate themselves into many different protein networks and possibly find one where they can be of use to the cell. On the other hand, as genes get older, they'll start binding with fewer and fewer things, but they'll be more selective about what they bind with. They'll probably bind with them more tightly. We get a much different shape of a graph when we look at aggregation propensity. So if you translate intergenic DNA, you'll tend to get a lot of proteins that create tangles or plaques that are toxic to the cell. Whereas the newest genes are already very different from the translated intergenic DNA, because you're not going to become a gene that actually codes for a protein if you're not useful to the cell and yet you're making these toxic plaques. So the newest genes, in fact, have even lower aggregation propensity than older genes. So how do novel protein structures and functions appear in genomes? Basically, we suggested that the 3 gigabase pairs of intergenic DNA that humans are carrying around can be a reservoir for a novel sequence, and many new genes will be created with that. These genes will behave nicely or be innocuous inside the cell while they're trying to join gene networks, and while most of them will be lost, a few survivors can be a source of novel domains, and maybe even of novel function. So genes are really acting sort of like tech startup companies. You may create a lot of them cheaply. They don't have a business plan. The ones that spam their users all the time will quickly be lost, but some of them can actually provide new services for their customers, unlike the startups that just copy old companies. Thank you for listening, and I hope you'll come to my poster tomorrow, L02.